So the EBV paradigm in marrow transplants is one which is really very problematic. About 30 to 60% of patients who undergo a marrow transplant for myelomas or other diseases like leukemia will actually activate with this virus. And there's a proportion of these patients who, by virtue of the fact that they're very immunosuppressed, will develop an EBV lymphoproliferative disorder. It's about 1 to 3% of unmodified grafts, and it's about up to 3 to 5% for cord bloods, and T-depleted grafts, it's even 5 to 7%. And the critical variable here is that these are monoclonal, usually diffuse large B-cell lymphomas. They are extremely aggressive, and normally the lifespan for these patients has been measured at less than 31 days with conventional treatment. They present with fever, exudative pharyngitis, or they can have lesions in the gut or in the brain. The limitation here has been that we know since 1995, when we did this initially, you can administer immune T cells from the transplant donor, and they can in fact induce durable remissions of disease. And since then, several groups in Europe, and particularly also the group at Baylor, have also shown that EBV-derived or EBV-specific T cells generated from the transplant donor can be used to effectively deal with these disorders. The limitation that we have found is that, unfortunately, it takes about 60 days or more to actually generate these T cells. And so what we wanted to do was to develop an approach which would allow for the development of these T cells and the use of these T cells in the rapid order which is really required because, again, the median survival is only 31 days. So what we developed here is a bank of 330 transplant donor-derived EBV-specific T cell lines, and each of these has been consented specifically for the use in an individual other than the individual to whom they gave their transplant. They're all defined in terms of their HLA typing. They're also defined in terms of their EBV specificity and their HLA restriction. And they're demonstrated to lack any alloreactivity that would cause graft versus host disease, and these are actually usable then off the shelf and can be given to patients within 24 hours of finding out about the disease. An important question is, how useful is this bank? And what we did was to survey over 200 unmodified marrow transplants or T-depleted grafts, as well as 100 cord blood grafts. And what we found was that we were able to identify a suitably matched, that is, 2 allele matched for HLA, and appropriately HLA restricted population for, as you see, over 99% of individuals. And in fact, for the group that we've looked at, we've had 175 requests. We've actually been able to find donors for 98.6% of these. Now, I'm going to report on 57 patients. As you see, there are 26 patients who received transplants or T cells from their transplant donor, and the rest of these individuals, the 31 individuals who received them from the third-party bank. The critical point to be raised here is that these are patients who have received beforehand rituximab and had failed Rituxan. As you can see, this is particularly the case for the third-party cases, including all the patients in the protocol 11130. You also note that these are almost exclusively large B-cell lymphomas. These are, again, clonal. We have only 5 patients who received T cells from the transplant donor who had viremia only. All the rest of the patients had pathologically documented disease. The treatment course is shown here. What we do is to administer weekly doses of 1 million or 2 million. The first trial was with 1 million. The second was with 2 million per kilogram of EBV-specific T cells. They're infused over 5 minutes by intravenous infusion. We give this once a week for 3 weeks. We wait for 3 weeks to look at them originally, specifically to determine whether there was any GVH potential. And if a patient has a partial response or a complete response, they would not receive anything more. Partial responses, they could receive additional dosings, or in some instances, as I'll show you, you could use alternative donors as well. As you can see here, in terms of the primary donor population, 62% had a CR and PR. In the third-party group, if you look at the combined story, it was 54% in the first study, 67% in the second study, and an overall 61%. The critical point that is different is that when you give transplant donor-derived T cells, these cells have the capacity to engraft, they will expand, and they will stay expanded and may stay around for periods of upwards of 10 years. In the third-party cells, these cells do not durably engraft. They will stay around for periods of anywhere from 14 to 28 or up to 90 days. But as a consequence, if you give repeated courses of these cells, you have the capacity to sustain the immune response against EBV. And so as you can see over the course of this, the CR and PR rate that we can achieve with the third-party cells is exactly the same as what we achieve with the transplant donor-derived T populations. And usually we will see these responses by the second or third course of T cells. Now, this is also important from the standpoint of dealing with the patients who have unusual typings, and we chose this slide specifically for the following reason. EBV is a virus which has an extraordinary capacity to mutate. And there are instances in which you can demonstrate T cells of donor type generated against the lab strain of EBV that are incapable of reacting against the EBV lymphoma that is in the patient. The basis for this has been documented in certain cases to be due to, in fact, deletion of whole epitopes or mutations in those epitopes such that the T cells can't see them. Now, the third-party T cell allows you to do something else. And this is shown in this slide. As you can see, this is an individual who developed really quite massive disease that you can see down below in the intestinal tract and otherwise. And in the red, what you're looking at there is the LDH. In the blue, you're looking at cytolytic precursors. And what we observed was with the first donor whose T cells were restricted by HLA-A1101 and the second donor, which was A1101 and 2402, we did not see any alteration in the course of the disease. The patient continued to have progression throughout the intestinal tract, but these were restricted by the A1101 and 2401. In contrast, when we took a third donor, line D, which is restricted by a separate HLA restriction, and therefore these T cells would be specific for a different epitope, we give those, and you can see in blue the expansion of the CTL precursors in vivo and complete resolution of the disease. This patient is now a year and a half out, has not recurred with evidence of the disease. So overall, if we looked at particularly the patients who are treated for rituximab refractory disease, you can see for the third-party donor populations in combined, the CR plus PR rate is 63%. And this is quite a striking feature. If we look at the long-term survival of these patients, you also see that in the first course, or the first trial, the third-party donors have a 50% long-term disease-free survival. And it's important to note that of the whole group of patients treated on this first protocol, 40% of them are alive up to 18 years post-treatment. As you can see in the second grouping, the disease-free survival is almost 72%. So what we can suggest here is that adoptive cell therapy with third-party T cells has been quite safe. It's been associated with very minimal toxicities. We've not had grade 2 acute or chronic graft-versus-host disease. We have not seen the cytokine release syndrome seen with the CAR cells. Second, these are available off the shelf, which means that these cells can, in fact, literally have a 24-hour turnaround from time of readout in terms of HLA typing, search, and returning the cells. They're efficacious in terms of durable CRs and PRs, and they have received this breakthrough designation. The bank permits selection of appropriate cell lines based on HLA partial matching, and in particular, HLA restriction. The bank also allows you to choose other donors of a different restriction shared by the patient's disease. And the basis for this is the idea that if you choose a second epitope, the likelihood that that would be mutated will be low. And we have shown that this is an effective approach in 5 of 7 patients that we treated thus far. They may require certainly more than one cycle, but the cycles are well tolerated. And with the achievement of the CR, the critical variable is these are durable remissions for multiple years, and similarly, intriguingly enough, different from the chemotherapy paradigm, the PRs are also durable in these patients for periods of years. And this is thanks to the group at Memorial Sloan Kettering Cancer Center that has put this together, and particularly to Sue Prokop, who's going to present the talk later on today. I'll take any questions.
So I'll begin with the first of laboratories that can do this. Here you've presented an off-the-shelf reagent for a devastating complication of bone marrow transplantation. Could you please just explain to all of us how was it possible to develop off-the-shelf in this scenario, and why has it not been possible, for instance, with TILs for melanoma or T-cell receptor engineered T-cells for sarcoma, this sort of thing?
Well, in this one, there's history here in that when we initiated our trials with these EBV specific cells, we recognized the fact that the survival of these patients was really short. And so what we did was to say, if a patient is at risk, we will make a T-cell up front. That allowed us to make the bank. And once we characterized the bank, then the other question is, could it be more broadly applicable? My sense of this is that this is an approach. You can develop these banks which could potentially certainly deal with several different types of infection complicating transplants or other disorders associated with severe immunocompromise. In terms of the development of T cells for tumors, then the real question is the degree to which you can identify antigens on the tumors which are essential to the lifespan of the clonogenic cell population, and then use those. We're looking at several other groups are looking at PR1, WT1, a variety of other different antigens, and the question will come up, can these T cells actually be used? Now, the studies at Rosenberg, and he presented it elegantly yesterday, he's actually looked at the TILs and is basically finding patient-unique antigens. My continuing question and my quest would be to ask, is it possible that there are going to be recurring mutations where we can identify peptide antigens, develop these kinds of pools of cell populations, which could then be used for therapeutics? And my general sense of it is, and this part of this may be the fact that, I'm patient and I also am into the glass half full mode. My general sense is it's probably ultimately going to happen.
We'll now take questions from the audience. Go ahead.
Lynn Peterson with Trends in Medicine. So do I have it correct that this has been licensed to Atera Biotherapeutics, and so then if a patient gets sick, you send it away in the company, or do the hospitals have to store this, or how would this be done? At
the present time, it's not licensed. It's under option from Atera. Right now, the bank that we have is fully open. That's where we are at the present time.
So a hospital in Texas contacts you if they have a patient?
Which they do. And right now they will send...
Can they? I mean, this is what...
Yes.
Logistically, how is this going to work nationally?
Right now, at the present time, what we have is a circumstance where we're treating them patients at Memorial Sloan Kettering Cancer Center. But what we're working towards is the idea that we would have groups where, in fact, we would be able to ultimately be in a situation where they would send the HLA pathology, we send the cells back by return mail. That's the intent.
Go ahead, next question.
Thanks. Yeah, Peter Droppert, Biotech Strategy blog. Could you take a step back and put this in the context of clinical practice of stem cell transplants in the United States? So I talked to other transplanters who said they didn't see sort of EBV lymphoproliferative disease because they had different transplant protocols, but what was taking place, or maybe what your approach at Memorial was more related to what your approach was, which was related to T-cell depletion in how you do transplants. So, is this very much an approach that is geared as a result of that transplant approach, and therefore, what you're seeing is again, in rituximab refractory patients, very good results, but in a very, very small subset of patients, but we're going to have to look at really case by transplant center how they do that, and would it be better actually to address modified approaches to transplant, which others are obviously doing clinical trials about?
Well, unmodified, the unmodified marrow transplant, as I said, is associated with 1% to 3%. It depends on the type of drugs or agents that you use to try to prophylax. For example, if you use certain monoclonal antibodies in vivo, you can get a rate up to 27%. That's been documented, for example, in the Seattle series early on. Unmodified grafts, because you're transferring T cells, have a lower likelihood because of the fact you do transfer EBV-specific T cells in the graft. You also have the issue of graft versus host disease. The T-depleted graft reduces the risk of GVH. It does put one at a higher risk of EBV. The cord T cells are naive to EBV, and the incidence of 3 to 5% is really quite stable. My sense of this circumstance right now is that we have ongoing national trials comparing T-depleted versus, for example, post-transplant cyclophosphamide, and I'm very interested to see on that because I think the incidence of GVH will be markedly lower and the overall long-term survival will be really quite good, but that's just totally separate from this particular talk. I think that the unmodified graft, nevertheless, has been associated with a very high incidence of acute and chronic graft versus host disease, which can lead to lethality in up to 15% of the patients at least, and it can be a really bad chronic problem. And so each group is trying to develop new approaches which would suppress the patient's the graft's capacity to reject the patient, and the limitation you have there is that many of those are associated with EBV. And nevertheless, any way you cut it, it's a small group of patients who get this particular complication. The real problem is if you're going to do that for the transplant donor, it's a very, very difficult circumstance to do in the context of time. What this means is that you've got cells off the shelf that can be used pretty much by anyone.
Okay, any other questions? If not, we'll conclude this morning's press conference presentation. We'd like to thank